Um, let's see, continue. Okay, so I'll talk to you today about my journey with psychedelics, which began almost 25 years ago in Nashville, Tennessee. But first, a little bit about psychedelics. Psychedelics first came from nature, and some examples of that are shown in this slide. Dimethoxytryptamine is a potent psychedelic found in the vine Psychotria viridis, which is part of the brew ayahuasca, which is used in South and Central America. In the desert Southwest, the peyote cactus contains mescaline, which is a psychedelic. And also in the Southwest is the Colorado river toad, whose secretions contain 5-methoxy-DMT, which is a very potent psychedelic. And certain species of mushrooms contain psilocybin, shown here as Psilocybe cubensis. But you don't have to go to the jungle, you don't have to go to the forest to find psychedelics. All you have to do is go to your local Home Depot or your neighbor's yard and look at their decorative lawn grass, which actually contains a fairly decent amount of DMT. So the first psychedelic that was actually purified was mescaline. It was identified in 1897 by the German chemist Arthur Hefter. And what he did was to fractionate the peyote cactus and individually ingest each fraction until he found the one that had the psychoactive effects. And this was mescaline. It was known, it wasn't very widely used, not very potent drug. It induces nausea in the people that take it and vomiting. LSD, or lysergic acid diethylamide, is probably the most infamous psychedelic. It was first synthesized in 1938 by the Swiss chemist Albert Hofmann. He didn't really find anything remarkable about it, let it go for a while, and then 5 years later, he had some kind of premonition that he missed something about this particular chemical from 5 years earlier. So he went back into the lab in 1943 and resynthesized a batch of this LSD molecule. And later that day, he started having some very mild feelings, some psychedelic effects, and he speculated it was due to the chemical that he had been synthesizing that day. So to validate this, he went back a few days later on April 19th and took what he thought would be a threshold dose of LSD, 0.25 milligrams. Drugs weren't really very potent back then. So 250 micrograms, he thought, threshold. But we now know that 250 micrograms is really a pretty large whopping dose of LSD. And he started to, after an hour, essentially have severe sensory distortions, hallucinations, and his assistant helped him home by bicycle that day. And now April 19th is usually celebrated as Bicycle Day in certain circles. So LSD, the hallucinogenic properties were discovered in 1943. Not too long after that, the structure of serotonin, 5-hydroxytryptamine, was identified. And a couple years after that, serotonin was found in the brain. And in the mid-1950s, the ability of LSD to induce what was then called a psychomimetic state, along with the discovery of serotonin in the brain, led Woolley and Shaw in 1954 to propose that mental illness, such as psychosis and schizophrenia, whose symptoms resembled the effects of LSD, were actually due to something involving the serotonin system. And this was really the first recognition that mental illness had a biological basis, that mental illness wasn't really purely Freudian, that it was biological. So the near contemporaneous discovery of LSD's properties along with serotonin in the brain really were the beginning of modern psychopharmacology. So the structure of LSD and other psychedelics all have similarity to 5-hydroxytryptamine. So serotonin is really a conductor of brain activity. It's everywhere in the brain. It mostly enhances or slows communication between brain cells and brain circuits and coordinates communication. And I like to think of serotonin really as kind of the conductor of an orchestra that facilitates the communication between areas of the brain, telling them when to become louder, quieter, stronger, or less communication. There are 14 different primary serotonin receptors in mammals. There are several more splice isoforms and edited isoforms that serotonin interacts with to exert its control. And all of these are expressed in the brain, although only subsets of certain serotonin receptors are found in the periphery. Now, psychedelics in particular activate only one receptor type to produce their effects, and that is the serotonin 2A type receptor. Studies in animals, studies in humans have shown that serotonin 2A receptor activation is necessary and sufficient to produce the psychoactive and psychedelic effects. There's high expression in several cortical areas, including the frontal cortex. So when I started my postdoc at Vanderbilt in 1997, we were asking, what is LSD doing in the brain? And is it related to schizophrenia? Because at this point, in the mid-90s, there were probably less than about 10 people in the whole world who were actually working on preclinical research with psychedelics. There was no clinical research being done. And the work that was being done with LSD and other related drugs was really in the framework of, can it be used to model psychosis and help to understand psychosis and schizophrenia? So my first project as a postdoc at Vanderbilt was really analyzing the molecular and genetic response to LSD in the brain to see if it could give us clues into what schizophrenia and psychosis was. So we performed the first unbiased genomic screen of the effect of a psychedelic in the brain. And yes, we did this screening at the University of Tennessee, Memphis Microarray Core. We had, in 3 separate rounds of screening, the Affymetrix rat A, B, and C chips were screened. And the results of these were published in 2002 and 2004. So I have nothing but good things to say about the microarray core as it was at UT Memphis. They were very helpful. Altogether in these 3 gene chip screens, the software only differentially called 60 genes as differentially expressed. And I systematically went through each of these, validating first by RNase protection and then by qPCR to really narrow it down to only 12 genes total that I could really find from these screens that were differentially regulated by LSD. So a very small number, and there were no genes that were downregulated. They were all upregulated. But surprisingly, or maybe not surprisingly, most of these were involved in synaptic plasticity. There were no genes that indicated any kind of toxicity or cell death, even though we used a very large dose of LSD with these rats. The area of the brain we looked at was the prefrontal cortex. So this is some data from the second 2004 paper where we looked at expression profiles. We did several dose response experiments. We found that there were 2 different dose response profiles. We did time course experiments. We noticed that there were 3 different time courses of response over 5 hours that we tested. Most genes were back down to baseline by 5 hours, with the exception of 1, which was NOR1, which stayed maximally influenced. So by this state, we realized that LSD, at least at the genetic and genomic level in the brain, was not doing anything harmful or dangerous, and in fact, may be doing something beneficial by activating genes that were involved in synaptic plasticity. So I moved to LSU Health Sciences Center as an assistant professor in 2004. And at this time, I was collaborating with my father, Dr. David Nichols, who is at Purdue University, who is also in psychedelic drug research. And what he had noticed in experiments doing drug discrimination with the LSD trained rats, and these were rats that were given a low dose of LSD 3 times a week to maintain their behaviors, that they developed these strange aberrant behaviors. They became hyperactive, reactive, they had sleep disturbances, they were over-aggressive, they had anhedonia, and a lot of these behaviors were similar to rodent models of psychosis and schizophrenia. So we wondered if the chronic LSD dosing was producing some sort of neuroadaptation that was relevant to psychosis and schizophrenia. And the interesting thing about this was the behaviors began about 6 weeks after treatment and stabilized at 3 months. And then when the drug was discontinued, so no more LSD on board, these rats remained just as abnormal for the several months that they tested. So there's some possibly permanent neuroadaptation that was going on. So my father generated 2 cohorts, a control and an LSD treated for 3 months, and then validated the abnormal behaviors. And then I flew up to West Lafayette and pulled the brains from these animals, brought them back down to New Orleans, and dissected out the medial prefrontal cortex. As you can see at the bottom of the slide here, this is a GTP gamma S activation of a coronal section. The medial prefrontal cortex is the most highly activated area in the brain by LSD. So we pulled this out, purified the RNA, and at this point, we sent it to the Vanderbilt sequencing corps in 2011, and they performed the sequencing for us. But at that time, the algorithms for the bioinformatics for a project like this, where we had 2 groups of 10 animals each, so 20 different rats each compared to each other independently, the algorithms really weren't quite ready for prime time to do this sort of analysis. So my graduate student at the time, David Martin, undertook a heroic effort to really dive into the literature, teach himself bioinformatics. We started a collaboration with Galaxy. They gave us some extra space on their servers, worked back and forth with us. And eventually we came up with a pipeline that gave us some very reliable data sets. And this was published in 2014. And David's analysis pipeline is shown here, primarily using Galaxy for the alignments and the mapping, HT count and DE-seq for the differential expression, and then functional clustering and validation. So we actually found several hundred genes that were differentially expressed. Most of these genes were about 25 to 50% changed in expression. We didn't find any genes that were twofold or greater. So what that tells us is that this suite of several abnormal persistent behaviors really was the result of several genes interacting and each gene having a low effect in that. To validate our data set, we selected 25 randomly selected genes and validated about 70% of them as being differentially expressed. And we found a very good correlation between the amount of expression found by the RNA-seq and by the qPCR. So at this point, we were fairly confident that we had a fairly good data set of what was going on in the brains of these rats 4 weeks after the last dose of LSD. So we did a lot of functional clustering. An example of one of the analysis we did is shown here. We did a string analysis, and we did see some unusual clusters of genes. For example, circadian rhythm genes, RNA processing genes influenced by LSD, cell cycle related genes influenced by LSD. But overall, in our final analysis, we saw that there was over a 3-fold enrichment for genes that had been implicated in schizophrenia by others, and that this was highly significant. So our hypothesis really was validated in that this model really was doing something that was relevant to understanding psychosis and schizophrenia. So that was 2014. 2015, 2016, really, that's sort of when the roof blew off of the whole field. That's when studies were published showing that psychedelics have therapeutic effects for psychiatric disorders. And one of these hallmark studies was by Roland Griffiths at Johns Hopkins University, who demonstrated that a single dose, a single treatment of psilocybin had long-lasting, up to at least 6 months effect to basically treat depression without remission. There was a recent follow-up study by the NYU group who showed that on average, 4.5 years later, 70% or so of the patients who responded to the single dose of psilocybin still had not had a relapse into major depression. So a single dose of psilocybin in a therapeutic environment can treat depression effectively for several years in some cases. We don't know the basis for that. I'll try to touch on that a bit in my research later on. Also, psilocybin has been looked at for alcohol dependence, smoking cessation, several other psychiatric diseases. I think there are now over 30 registered clinical trials with different psychedelics now at clinicaltrials.gov. Around the same time, imaging and network-based studies were also being performed with titles such as Dreamlike Effects of LSD on Waking Imagery, Effective Connectivity Changes in LSD-Induced Altered States of Consciousness, Increased Global Functional Connectivity Correlates with LSD-Induced Ego Dissolution. So around the same time, these imaging and network studies were coming out. So what we knew at that point were that psychedelics induce altered behavioral states by activating the serotonin 2A receptor in the brain. PET scans had shown that areas like the prefrontal cortex had increased metabolic activity. And fMRI scans showed there was altered bold activity also in certain areas like the prefrontal cortex. High-resolution fMRI scans were then performed to look at network connectivity. And this publication was a key publication from David Nutt and Robin Carhart Harris's group at Imperial College in London. And this is some of their data on functional connectivity. And this circle plot represents the different brain areas. Each little circle here would be a different region of the brain. And the line connecting the circle represents those that are connected. And the thickness of the line is the strength of that connection. And this particular connectivity plot is the default mode network. So that's the state of connectivity that your brain is in when you're not really doing anything. Daydreaming, zoning out, really no processing going on. If you then image after somebody has taken psilocybin and is at the peak of the psilocybin experience, you can see that there's this massive functional connectivity change within the brain. There are some areas that are decreased in connectivity, other areas that are increased, but overall, there's an increase in functional connectivity. So you have a lot of brain areas that normally aren't talking to each other and now talking to each other. So based on the imaging studies, we now have a fairly good idea of how psychedelics alter communication between brain regions at the network level, in that they produce a disintegration of the default mode network and increase connectivity. And there's a theory that in the treatment of depression, what this does is when the connectivity resets back to the default mode network state, that it resets to a more normal non-pathological state, although that's just a hypothesis. So I think the most valid hypothesis now for the overall psychedelic effects is the revised gating hypothesis from proposed by Franz Vollenweider and Mark Geyer. And this is taken from a paper from Franz Vollenweider and Katrin Preller from earlier this year. And it involves the thalamus and its connections to specific cortical areas. And what they have proposed is that through activation of 5-HT2A receptors on the thalamus, that there's a reduction of thalamic filtering and filtering of interoceptive and exteroceptive stimuli, and that there's decreased connectivity between association cortices and the rest of the brain, which leads to decreased integrative processing. But there's simultaneously an increased connectivity between the sensory cortices and the rest of the brain, leading to increased sensory processing. So there's this imbalance of increased sensory input, increased sensory processing, but decreased integrative processing of this sensory information that together leads to the psychedelic experience. But network analysis and clinical studies are really only part of the picture. That clinical studies inform on potential therapeutic strategies, like what can we use, what can we treat psychedelics with, but they don't depend on knowledge of mechanism, and they don't inform on knowledge of mechanism. Imaging studies are, by and large, primarily observational, informing on fundamental brain network function, and telling us what brain networks underlie specific aspects of normal consciousness and perception. And then using psychedelics as tools, we can alter the normal function in specific ways relevant to understanding certain cognitive processes and the brain connectivity underlying these processes. But neither the clinical or the imaging studies really inform on how psychedelics are interacting with their target receptors, or influencing cellular biology that drives the observed network changes underlying the therapeutic effect. And this has really been the core of my research since I started back as a postdoc, is to understand the molecular and cell biological mechanisms of psychedelics. And understanding this is vital for further developing psychedelics for medicines in the clinic to treat not just psychiatric disorders, but other disorders. Okay. So onto the brain. There's a high receptor density in layer 5 of the cortex of serotonin 2A receptors. So this is a rat brain coronal section of the medial prefrontal cortex from our lab, stained with antibodies for the 5-HT2A receptor. And the receptor itself is primarily expressed on the cell bodies and the apical dendrites of the glutamatergic excitatory pyramidal cells. It's at high levels on these cells, but it's at low levels on several other types of cells in the brain. Okay, so now we take a deeper dive to the receptor in the membrane. And this is a space-filling representation of the 5-HT2A receptor, a molecular representation, and then a ribbon representation. The serotonin 2A receptor is a G protein coupled receptor. And this is shown with LSD in the binding pocket. The interesting thing about this, and the paper on the crystal structure of LSD in the 5-HT2A paper in the receptor just came out a couple weeks ago from Brian Roth's lab. My father was a co-author on that paper. The interesting thing about LSD is this extracellular loop 2, that when LSD binds to the receptor, it comes down and folds a lid and keeps the drug trapped in the receptor. So the receptor has the drug in there and it's signaling for 8 hours or more. Other psychedelics like psilocin or DMT, they'll pop onto the receptor, they pop off. It's LSD that really gets stuck on there. So now we believe that that's why LSD is such a powerful and potent psychedelic compared to the others, because it gets trapped in the receptor and stays there signaling for several hours. So another aspect of the 5-HT2A receptor is functional selectivity. And that is that psychedelics activate the receptor differently than serotonin. I'm going to try to illustrate that with this slide. We have the receptor on the membrane. When serotonin binds, it activates several pathways, but the canonical pathway is the G-alpha Q pathway that activates phospholipase C, producing mannitol phosphates and calcium. When psychedelics bind to and activate the receptor, it's thought that they change the conformation of that receptor so that it recruits other additional signaling pathways that may not be as normally strongly recruited to the receptor with serotonin. One example is the G-alpha I pathway. And a third pathway that we're trying to elucidate is we discovered several years ago that psychedelics are anti-inflammatory. And this does not involve G protein signaling or beta-arrestin signaling. Okay, so again, back to the brain. We have looked at where the 5-HT2A receptors are, but we can also look to see what the cells are that are activated by psychedelics. In this case, we treated rats with the drug called DOI, 2,5-dimethoxy-4-iodoamphetamine. It's related in structure to mescaline, but it's highly selective for the 5-HT2 receptor. We can't, or I don't really like to use LSD for these types of studies because it's a very dirty drug. It hits almost all serotonin receptors, dopamine receptors, adrenergic receptors. So we get the cleanest results using a 5-HT2 receptor selective antibody. Although we do see very, very similar, almost identical results comparing LSD to DOI. So the green dots here represent cells that have become activated in response to psychedelics as stained by the immediate early gene CFOS. You see there's an enrichment in layer 5. But at Vanderbilt, when I was there, both in Elaine Sanders Bush lab and in Ariel Deutsch's lab, they had noticed something that we also did, that the cells that become activated in response to psychedelics aren't the cells that express the receptor. Here you can see the receptor expressing cells are circled in red, and the activated cells, the green triangles. We can block the activation using a 5-HT2A receptor selective antagonist. So we don't really quite understand why the cells that are becoming activated, at least transcriptionally with CFOS, are those that don't appear to express the receptor. So this was kind of a conundrum, and it sat in the back of my head for years. Nobody really paid much attention to it. And then after David finished the bioinformatics project, we started to work on this and to figure out why this was. Why don't psychedelics appear to activate the neurons that their target receptor is highly expressed in? So when we did further analysis with some confocal microscopy and more detailed analysis and staining, we found that there were some small percentages of cells that were staining for both CFOS and 5-HT2A receptor. So they are there, they're just very rare. So my hypothesis at this point was that there's a subpopulation of neurons in the cortex expressing 5-HT2A receptors that directly respond to psychedelics and are activated by psychedelics. And that these cells represent really the trigger for the overall effects that initiate and produce the trip. And that these play a key role in modulating normal cognitive processes. So I called this cellular ensemble the trigger population. So our next step was to go after these cells, to purify them from the brain and begin to molecularly characterize them and see why they are different than the others. To do this, we developed new methodologies of flow cytometry, and we called this neurocytometry. And as opposed to previous methods, which you had to use either a genetically tagged nuclei or fresh tissue, we developed protocols where we could use fresh brain tissue, frozen brain tissue, fixed tissue, frozen fixed tissue, and even routinely fixed human postmortem brain that maintained cellular integrity for the extracellular membrane, the cytoplasm, and the nucleus. So we weren't just purifying nuclei. And here's an example micrograph from one of our neuronal sorts, and you can see that not only do we have a cytoplasm, but we also have some cellular processes that are still there. So if you want to read more about this, this was published in ACS Chemical Neuroscience a few years ago. So our pipeline was to treat the animals, take the brains, and we looked at the medial prefrontal cortex as well as other brain areas, perform our neurocytometry, and gate so that we're purifying out the activated, the cells that are activated by psychedelics. This is an example of some of our sorts. On the left is a sort of a brain where we're separating the neurons from the non-neurons using this nuclear marker, NeuN. This top cluster are neurons, the bottom cluster are non-neurons. And on the x-axis is CFOS. And on the right panel is a brain treated with DOI. And you can see that there's a slight number of cells with increased FOS expression in these DOI treated animals. And altogether, it's about 5% of neurons are directly activated by psychedelics. And this was a pretty large dose of DOI, so a fairly small number of neurons are being directly activated. So we sorted these out. And we also sorted out the bottom 80%. And so we had two different populations. The psychedelic activated cells, which are represented by the FOS positive, and the non-responding cells, the FOS negative. And when we looked at these cells for the mRNA for the 5-HT2A receptor, we saw that the activated cells have about a 4-fold increase in the expression of the mRNA for this receptor. So we believe that the trigger population neurons are more sensitive to the effects of not only psychedelics, but also serotonin due to increased receptors. We also looked at other genes that were implicated in psychedelic effects. One of them is the glutamate 2 receptor, the mGluR2. And we looked in the medial prefrontal cortex, again, which is a central key area for the mechanism of psychedelics. And we also looked in the somatosensory cortex, which is on the exterior of the brain. A lot of studies use somatosensory cortex rather than medial prefrontal cortex, I think because it's potentially easier to get to and it's larger. But our results really underscore that the neurons within these two brain areas, at least at the molecular level, are different, and they have different responses to psychedelics. For example, mGluR2 is enriched in the activated neurons in somatosensory cortex, but it's not enriched in activated neurons of the medial prefrontal cortex. We also looked at GABAergic inhibitory interneurons, and we looked at the 4 different subtypes: the parvalbumin, the somatostatin, calretinin, and the 5-HT3 GABAergic interneurons. And we found that of those 4 subtypes, 2 of them were directly activated by psychedelics. This shows the results for parvalbumin, and you can see that parvalbumin interneurons are activated in the medial prefrontal cortex and in the somatosensory cortex. For the somatostatin interneurons, however, there's no significant activation in the medial prefrontal cortex, but there's a very high activation, almost 20-fold activation in the somatosensory cortex. The conclusions from this work were that about 5% of 5-HT2A receptor expressing neurons become activated, and I called these the trigger population. They have higher expression of 5-HT2A receptor mRNA, and I'm assuming that they also have a higher receptor protein expression, but we haven't tested that yet. Only neurons in the somatosensory cortex and not the medial prefrontal cortex that are activated by DOI are enriched with mGluR2. We see about 10% activation of GABAergic interneurons, but these are only the parvalbumin and somatostatin subtypes. I don't know what roles these are playing. That's something I'd like to explore in future experiments. And we also know that glia and astrocytes, about 10%, become activated as well. And I'm particularly interested in knowing what astrocytes are doing and if they're playing a role in the mechanism of action of psychedelics. So you can't really think of psychedelics as activating or inactivating a particular brain region when thinking about its connectivity. You really have to get more granular than that and think about psychedelics activating subsets of excitatory, inhibitory, and non-neuronal cells that really govern the activity of a particular brain region. And that this heterogeneity between the psychedelic response, I think, underlies the heterogeneity in the network connectivity changes that are seen in the imaging studies. So this research was published. Psychedelics recruit multiple cellular types and produce complex transcriptional responses with the brain, also by David A. Martin. He is now a postdoc at University of Baltimore in Maryland. University of Maryland, Baltimore. So what are the consequences of these molecular effects? Well, several years ago, when David was in the lab, he did some pilot experiments looking at Golgi stains of pyramidal neurons in the medial prefrontal cortex. And these are dendrites of glutamatergic pyramidal neurons in the MPFC. This is a control brain. On the left and on the right is one that was treated with LSD. And we found that a single treatment with LSD significantly increased the synaptic density of the medial prefrontal cortex excitatory neurons. We also looked in the fruit fly brains, and we looked in the glutamatergic neurons in the central fly brain using a GAL4 V-glut driver to label the ellipsoid body glutamatergic neurons with GFP. We treated the flies with LSD for 24 hours and then looked to see what effect that had on the ellipsoid body. And the ellipsoid body is a complex structure in the fly brain that receives sensory input and has motor output. And we looked at not only the size of the ellipsoid body, but also what we call the energy. And that represents what the texture of the pattern is within the ellipsoid body glutamatergic neurons. And the texture became more complicated, the size became larger. So together, this tells us that LSD in the fly also increases synaptic density in glutamatergic neurons. So we never published this data, but a couple years ago, David Olson at University of California, Davis published some really exciting results looking at cortical cells in culture with psychedelics, and also with Drosophila. I think he used the larval muscle, the larval muscle wall, and saw that there were increased synaptic density from several different psychedelics. Then what he also showed was that this synaptic increase was dependent upon mTOR pathways. So at this point, the questions were, how does a single administration of a drug lead to persistent therapeutic effects? You give a psilocybin to somebody once who's depressed, and 4 years later, they still haven't remitted back into depression. Are the antidepressant effects dependent upon the subjective peak experience? Is it purely a psychological effect, or is it a biological effect? And we needed animal experimental systems to answer these questions. So we set out to do that and developed 2 rodent-based models that we could study this in. The first that we developed was using this Wistar Kyoto WKY strain of rat. And this has been used historically as an endogenous model of depression. That this is a rat that normally it's born depressed. And if you treat it with certain antidepressants, it begins to start acting like a normal rat in certain paradigms. For a more translationally relevant model system, we took female Sprague Dawley rats and had a unique adolescent chronic restraint stress paradigm that was developed by Dr. Megan Hibicki, who's a postdoc in my lab when she was a graduate student at Mercer University. And this adolescent chronic restraint stress elicits lifelong deficits in behaviors that are relevant to modeling depression. So most models of depression in rodents are only effective in males. They're not effective in females. So it was important for us to show that that was effective in females, because in the human demographic, females are much more vulnerable to depression than males are. And further, typical stress paradigms in rodents, the depression self-resolves after a few days. So we really couldn't test the long-lasting effects of psilocybin. So in these 2 models, we gave them a single dose of psilocybin and then tested by various outcome measures that I'll talk about in slides that are coming up. So, okay, this slide shows results from our WKY rats, where we injected them with psilocybin on day 1 and essentially just let them hang out in their cage for 5 weeks until we tested them by the forced swim test and then by elevated plus maze. So the forced swim test is a model of helplessness, and normally a WKY rat or a rat that's exhibiting depressive-like behaviors will just float. And there's a very high immobility time. This assay has a very good predictive validity with respect to antidepressant medications in humans. This other rat here you can see on the right is a WKY rat, and it's one that's been injected with psilocybin. It's really behaving fairly normal. And when we look at the data grouped together, the immobility significantly drops in these rats that were given psilocybin. This effect size is about twice of what a typical antidepressant would be in the WKY rat. So essentially, these rats are behaving like normal rats would, not like depressed rats. So this was published in, I believe, February of this year in ACS Chemical Neuroscience. And we also looked at the effects of ketamine. Ketamine in this model only had antidepressant effects that we could detect out to about 2 weeks. And that's also very similar to what's seen in humans. Ketamine, a single infusion, has antidepressant effects for about 2 weeks until somebody needs another infusion. So we were really able to recapitulate the human demographic with this model demonstrating long-lasting persistent effects of a single treatment with psilocybin and more transient effects of ketamine. But there is questionable translational relevance of the WKY rat to humans because we don't really know what's making those rats depressed. So we moved to this chronic restraint stress. And in this, we took female rats during adolescence and essentially put them in a frosting bag for 1 hour a day for 2 weeks. And then in young adulthood, 1 week after the final restraint stress, we gave some of these rats a single treatment with psilocybin. And as our outcome measure, also the forced swim test has questionable translational relevance because we don't know what circuits mediate that. We used a defined neural circuitry test to look for object pattern separation. This is a behavior that is defective in depressed individuals and responds to antidepressant therapy. So object pattern separation involves placing a rat in an arena that has 2 identical objects, and on successive days, you move the object farther away from its initial plane, and normal rats will detect that one object has moved in relation to another object. And this is a cognitive assay. It's not a memory assay, and it's mediated specifically by entorhinal cortex, the dentate gyrus to the CA3. And there's similar pattern separation tests that are available in humans that have shown that the same circuitry is involved. So a high translational relevance of this cognitive-based outcome measure. The non-stressed animals, or control animals, had very good discrimination. They could detect when the object moved out of the plane. And this is data from day 5, where the object is moved farthest away from the plane. The stressed animals, these would be the depressed animals, the ones that received no treatment, they really couldn't discriminate pattern separation at all, really severely impaired. However, the ones that were treated with psilocybin performed just like normal. So the single dose of psilocybin normalized the dysfunction in this specific hippocampal circuit, and also we validated with the forced swim test, significantly reduced the immobility similar to the WKY rats. So very long-lasting, persistent, profound antidepressant effects in this more translationally relevant model. And if that weren't good enough, we've also looked at psilocybin and its antidepressant-like effects in Drosophila. So we identified a wild-type isolate, an isolate of a particular wild-type strain that had high immobility in the forced swim test. So we further validated and developed a forced swim test that was originally published by Wendy Neckemeyer at University of St. Louis. So in this, we found this strain of fly that was an isolate of the CS strain that had high immobility compared to another CS strain that we had in the laboratory. So we took this fly strain and treated it with citalopram when it was 2 days after eclosion for 5 days continuously. And we saw that this treatment with citalopram decreased the immobility, similar to an antidepressant would in the WKY or a traditional rat model. And we know that this isn't just an increase in locomotor activity because we did photobeam break activity assays with this strain, and citalopram really didn't have any effect on overt locomotor activity. So it's having antidepressant-like effects in this strain of fly. If we just fed these flies on citalopram for 1 day, tested them at 5 days later, there was really no measurable effect. Psilocybin, on the other hand, at a high dose and even a very low dose, were as or more effective as citalopram in this strain of fly. So psilocybin appears to be having long-lasting antidepressant-like effects in fruit flies as well as in rats. So this gives us several models that we can now begin to look at what are the molecular effects of psilocybin on different brain cells and brain circuits. So summary, antidepressant effects of a single treatment of psychedelics are long-lasting in rats and flies. And in data that I didn't show you, we found that the environment the rats experience in the days following the drug treatment really shape the overall antidepressant outcome. And so we believe that psychedelics really open a window of behavioral flexibility that allows the animal to develop better coping skills. And this recapitulates results that have been found in human clinical studies, and importantly provides us experimental systems that we're collecting tissues from for analysis. We've also demonstrated that psychedelics may be superior to ketamine in our models, that ketamine, as in humans, is only transiently active in the rats. And our overall conclusion so far, or our hypothesis at this point, are that the antidepressant effects in humans are likely biological in nature, and that while correlated to antidepressant effect, the peak ego dissolution, the trip, the subjective experiences of the individual under the influence of psilocybin are not causational to the antidepressant effect. Okay. So for the last 5 minutes, I'm going to talk about something completely different. About 12 years ago, or maybe more, we discovered that psychedelics are very potent anti-inflammatories. And the 5-HT2A receptor is the most highly expressed serotonin receptor throughout the body. It's expressed in nearly every tissue, every organ system. But unlike psychedelics, serotonin is primarily pro-inflammatory. When it activates the 2A receptor, there's an increase of pro-inflammatory biomarkers. One of the primary assays that we've used is allergic asthma. It involves several different aspects of immune system function, from mast cells, T cells, eosinophils, mucus overproduction, that together combine to produce significant inflammation and airways resistance. So we have generated mice and rats with asthma, treated them with psychedelics, either nebulized or injected, and then looked to see how that affected the inflammation in the lungs and their ability to breathe. This is a slide from one of our experiments of a control lung, and this is an airway in the control lung. See, it's fairly thin. This is a zoomed out version of the airway. This is an asthmatic lung. This is a mouse with asthma. It has very thickened airways. There's mucus overproduction, and this green staining is collagen. So there's fibrosis in this mouse. There's also a lot of inflammation that's present. If we treat these mice then with a psychedelic, again, DOI, but we've also used several other different kinds of psychedelics. Some are more effective than others. We can significantly reverse the fibrosis and eliminate the inflammation in this asthma model. And we can also restore the difficulty in breathing. And we test the breathing by doing 2 different types of protocols. One is a flex event, which is a forced oscillation, and the other is a whole body plethysmography. This is a forced ventilation. So we give these animals methacholine, and this is a muscarinic agonist that will cause a mild bronchial constriction. So the higher dose of methacholine, the more difficult it is, the more resistance there is. So this is more difficulty breathing. The asthmatic animals have this hyper response to methacholine. So they have a significant difficulty breathing at these higher doses of methacholine. When we give these animals DOI, we essentially bring this difficulty in breathing in the asthmatic animals back to normal baseline. So we're not only affecting the physiology at the lung level, but we're also affecting the ability of these animals to breathe again. So for a summary of anti-inflammatory, we have proof of principle preclinical data in several animal models, including vascular inflammation, atherosclerosis, metabolic dysfunction, like in diabetes. We can also have efficacy in inflammatory bowel disease models. We've done most of the work in asthma. A collaborator of mine, Dr. Tim Foster, has been doing a lot of work with ocular inflammation and shown some really profound, potent effects, anti-inflammatory effects in the eye. And key features of 5-HT2A agonist as as anti-inflammatory therapies are that they're really, really super potent. The levels of drug that we're talking about that are able to treat asthma, inflammatory bowel disease, are 30 to 100 times less than the minimal amount necessary to induce behaviors. So essentially, microdosing of our lead candidate DOI and several other types of psychedelics are effective therapies to treat inflammation. Also, these are steroid sparing. They're not acting as an immunosuppressant. If an animal is not sick, a psychedelic will not suppress pro-inflammatory biomarkers, so the immune system is still there, it's still present. So this would mean fewer side effects. And I'm working in partnership with a company called Ellucis Therapeutics to develop these molecules for the clinic, and hopefully we'll be in clinical trials in the not too distant future. So with that, I would like to acknowledge the various members of my laboratory over the years who have contributed to these projects. A lot of the work that I showed you was from David Martin, who was in the lab, who did all the bioinformatics and gene expression studies. Bengning Yu discovered the anti-inflammatory effects. And Thomas Flanagan is really doing all the asthma work now that was started by Felix Now, a former MD PhD. Megan Hibicki is doing all of the depression behavioral work. And Hannah Kramer is a graduate student in the lab who is now taking the brains from our chronically stressed animals, purifying the RNA, and we're going to be starting to do RNA-seq and ATAC-seq, looking at genetic and epigenetic changes that have occurred in key brain regions in the rats that have responded to the psychedelics. And hopefully we'll be able to work out some clues to the mechanism of action for the long-lasting therapeutic effects of psychedelics. As I mentioned, I'm working in partnership with Ellucis Therapeutics, and in full disclosure, I have a sponsored research agreement with Ellucis, and I am president of their scientific advisory board. Also, Betsy Gordon Foundation, NIH, Hefter Research Institute, LSU, and especially the American Asthma Foundation that really enabled a lot of the anti-inflammatory and asthma work to happen. So with that, I will stop and take questions.
Well, thank you. That was a great talk. And it looks like we have